Yeah, good afternoon. My name is Samuel from Texas A&M University. We all know that necrotic enteritis remain a major challenge in poultry production because it compromises gut health, bird welfare, and production, especially in antibiotic-free system. So today, I will be presenting our work on natural adsorbent as antibiotic-free strategies against necrotic enteritis. So poultry is a major source of affordable income and animal protein worldwide. However, necrotic enteritis caused by Clostridium perfringens damage the intestinal lining and compromises bird health. This leads to increased mortality, reduced weight gain, and poor feed efficiency, which cost the poultry industry over $6 billion annually. And also, due to the restriction of antibiotic growth promoters, this leads to an urgent need in antibiotic-free alternative for the control of necrotic enteritis. Now, this is what we try to achieve. We want to use this natural adsorbent, biochar, clay minerals, and graphite to mitigate this natural adsorbent, to mitigate necrotic enteritis. We selected this natural adsorbent due to their potential to bind harmful compounds such as net-big toxins, and also to reduce oxidative stress and increase cell viability. Our research objective is simple but critical. One, we want to evaluate the antioxidant, the antimicrobial, and barrier protective effects of natural adsorbent against Clostridium perfringens using chicken intestinal epithelial cells, and to test this adsorbent itself against this bacteria that is causing necrotic enteritis, which is Clostridium perfringens. To address this objective, we isolated primary intestinal epithelial cells from the duodenum, the jejunum, and ileum of 21-day-old chickens. We then evaluated the materials across the main endpoint, which is the oxidative stress, the epithelial viability, and the direct antimicrobial activity against C. perfringens itself. So this design allows us to assess whether this adsorbent could provide multifunctional protection in a biologically relevant in vitro model. So our first result is reactive oxygen species. As seen on my screen, across all the adsorbents tested, which is A, kaolinite, B, bentonite, C, Wyoming clay, and D, sodium morillonite, and E, graphite, F, wood biochar, and G, agricultural waste, we notice that the D, which is sodium morillonite, show a very good effect by reducing the oxidative stress compared to every other one. And also bentonite, graphite, and kaolinite also performed well across the adsorbent tested. Now, looking at the cell viability, in the cell viability, we next assess cell viability. The results showed high viability across intestinal concentration range, indicating minimal cytotoxic and supporting epithelial protection. In other words, these materials were not only active, but also compatible with intestinal epithelial cells under the study condition. Taken together, the antioxidant and viability findings suggest that selected adsorbent can help protect intestinal epithelial cells from injury-related stress while maintaining cell integrity. This is especially relevant in necrotic enteritis, where epithelial damage is a central feature of the diseases. Now, antibacterial. When we now assess the direct effect of this adsorbent against the Clostridium perfringens itself, then we noted that several adsorbents demonstrated a dose-dependent manner with high effectivity shown in high concentration. Just as you can see in the 100 milligram and 50 milligram, there is a high inhibition there. But as the dose reduced, so as the effectiveness of the natural adsorbent also reduced. And this is the microbial dilution method, which is also another method for assessing the antimicrobial effect. As shown on my screen, let's continue. This is a representative from the research as the C, which stands for MS Wyoming, E, which is the graphite. As you can see on the C, the 100 milligram, there is no growth. The only way where there is inhibition is the control, which we use penicillin as the control. While in graphite, we observe a zone of inhibition in 150 milligram, and as the dose reduced, the activity of the natural adsorbent reduce as well. And the X is the cell under 10x magnification of 75 to 80% cell confluency, which was used for the cell viability. And the Y is 90 to 100% cell confluency, which was used for the reactive oxygen species. Both of them were derived from the jejunum of 21-day-old chicken. Now, in conclusion, natural adsorbents reduce oxidative stress, support epithelial viability, and inhibited Clostridium perfringens growth. Among the tested material, sodium morolite showed the strong and most consistent overall performance. Overall, these findings support natural adsorbent as a promising antibiotic-free and welfare consciousness candidate for necrotic enteritis. Most importantly, this work also supports poultry welfare by using a primary cell-based model to reduce reliance on live animal experimental during early stage of screening. So instead of using live cells, we can use this in vitro method. Just a chicken is enough to test whether if this thing is effective or not. Acknowledgement. I would like to thank Mesla Boucher, Greensmith Graphite, Texas A&M AgriLife, and the Department of Texas of Poultry Science, Texas, for their contribution to make us to achieve this work. So thank you for listening. And this is my presentation. Thank you.